Seed germination, credit to C. Cone from Waterford, Wisconsin. Very good PowerPoint. Really suits our needs and really goes beyond what we need to know. So we'll highlight what you need to know, and if you want to differentiate and learn about other things, I'll offer you a bonus opportunity later. Seed germination is simply when the seed sprouts. So in your lab, you should see something today. When you look at your seed, what you're actually seeing is a seed coat. And the seed coat performs much of the same work that your own coat performs. It provides protection against parasites, diseases, viruses, etc. It protects against mechanical injury. That's like you get physical damage against the seed. Sometimes it protects you against high or low temperature. If the temperature is too high, it's really dry or hot, the plant will die. If it's really cold, the plant will die. Okay. And the seed will only open at the right time. Okay. So if it fails in the fall means like in August, September, October in America, because that's after winter or right before winter when you should have seeds sprouting. That's what they're talking about. It's in Wisconsin. Here are the different parts of the seed, and this is a very important slide as you should be aware of all the different parts of the seed. The cotyledon is like basically the seed coat. The endosperm is inside. Remember, endo means in. The radicle, hypothetical, and apical. We'll talk about what that turns into later. That's the embryo. That's a seed. Embryo means baby. And the seed coat is also part of cotyledon. Okay. Sometimes you have more than one cotyledon. We'll come up to that. That's a different type of plant. You have monocotyledons and you have dicotyledons. So some plants have one cotyledon and they have two cotyledons. And it leads to different types of leaf structure and a different type of plant. So you have your protected embryo, basically immature plant, important definition. You see leaves and roots. They're small leaves and roots. The leaves are the plumels. So think of the plum, like the fruit, they're the leaves. The leaves are sheeted by cotyledon. Sheeted means like covered. That's what that word means, covered. The embryonic roots are called the radicles. The radicles are the roots. And the stem is called the hypocotyl. So the important thing is the leaf is the plumose. They're covered by cotyledon. The roots are the radicals. And the stem is the hypocotyl. Okay. And the endosperm is the food. So that's like the yolk of the egg. And it forms the food for the plant to grow. See it again. Excuse me. So know what part the plumose, the leaf, hypocotyl is the stem, and the radicle is the root. So you should know what those parts are. And it looks kind of like a baby plant. Once you add water, it should force the seed to start the enzymes and it'll start the growth. And what the enzymes do is they break down the starch in endosperm, and they use that for energy. Now, the deciding factor is whether or not it's available for growth and cell division. So if you had your seeds and they're like tomato, cucumbers, you might have to dry them out. When you dry them out, it might have damaged the seed. If you have tangerines or watermelons, the seed coats might be really tough. And so just putting them in water and a paper towel, they might not grow. Other kinds of seeds, it really depends on the circumstances. But the first step is water. Here's the stages leading cell division. This is really complicated, and I'll explain it to you, but you're not responsible for everything in here. I'll highlight what you're responsible for. So you have your mitochondria. Reconstituted means like it starts to perform. Respiration means it's starting to breathe or grow or eat. Anaerobic is no oxygen. You should know that word. It's an important definition. Aerobic means you use air. So anaerobic is no air. Aerobic is air. Soluble sugars, we know soluble means dissolving. So ATP is energy to cell, adenosine triphosphate. The RNA is a ribonucleic acid that activates it. Polysomes are like another organelle within the cell. Then you have protein synthesis and enzymes. Enzymes are proteins that help reactions occur at a lower energy level. And then you have DNA synthesis, and then you finally have mitosis, which is cell division. Okay, So when you have your seed, you add the water, it pops up the mitochondria, starts producing energy, and it starts out anaerobic with no air. And the seed coat breaks, gets oxygen in there, starts to eat the sugar, and then it starts to grow. So the first thing is water. Seed dormancy. Dormancy means it's asleep. Okay, and so you have a waterproof seed coat, no oxygen because that seed coat's solid, and chemical inhibitors. An inhibitor means you're stopping the process. So like a person is inhibited means you're like shy. So the chemical inhibitors make the process slow down. For germination to occur, you gotta cancel out all these things. You must break down the seed coat so that your tangerines and your watermelon seeds, you got chip them. You would actually, so if you have tangerines or watermelon seeds, you probably have to take like a small little, be careful, knife and chip them. So you break the seed coat and then add water again to them in the paper towel, and you should see some growth by Friday. The water must be absorbed, and the growth promoters must be activated, the inhibitors must be inhibited. But once you add the water and just light and oxygen, it should work. The seed coat has to be penetrated first, though. These are the various stages of germination. You're not responsible for this page, but I want to throw it in here so you know the terminology. You're responsible to know pre-germination, germination, and post-germination, and very generally understand it, but it's very high level. Rehydration is when you get water back in. RNA and protein synthesis, that makes sense. Increased metabolism is how it eats. Hydrolysis is basically adding water to the system and breaking down the food by enzymes. Ultrastructure is just a structure. And induction of cell division and cell growth. Induction means it's starting. Okay, so pre-germination basically means you get the water, and then you have RNA synthesis, and it starts to grow. Germination is you see the seed coat break, and then you start seeing the seedling with the radicle first, which is the root. And then post-germination, you have controlled growth of the shoot axis, that's a stem, etc. And then you have transport of food, and then you have senescence, aging of food storage tissue. Senescence or aging means basically you digest the food, and once it's there, it becomes rubbish and disappears from the plant. On this page, you should know what pre-germination means, germination, post. And basically, pre-germination is when the water comes in, germination is the seed coat breaks, and post, it just starts to grow. That's your full requirement. However, if you want to go into more detail, you're more than happy to do that. Water is the most important thing. So if you didn't give enough water to your plant, it's not going to grow. It's a triggering enzyme for everything in the plant. Turgor pressure means you have a lot of water in the plant. So like when you have your lettuce and you're at the salad bar, that's why they always have lettuce wet, because lettuce will absorb the water and stay nice and crispy. Okay? If you leave lettuce out, it will just dry out as the water leaves and becomes very limp or flaccid. Flaccid means like soft. Turgor means like firm, full of water. Second factor besides water is light. You can either stimulate or inhibit cell growth. If you have too much light, it won't work. Some plants grow in the dark first, and this is how the seeds are going to be sown. Does it sprout on the surface, or do they grow underground where you don't need photosynthesis? So it depends on the type of seeds. Okay, I'm assuming the fruit seeds we have at home, watermelon, and the other seeds, you would not have to do that. It's specific to the seed, so I have to talk to you about your seeds. Oxygen is the third factor. You must have the plant must be able to breathe oxygen, and if you don't have oxygen, you can't remove the waste from the cell. And without oxygen, waste is not removed. Remember, in a plant, they breathe in carbon dioxide and they give off oxygen. So they need to have that during the day, but at night, and we'll learn this later, dark photosynthesis, they suck in oxygen, and it's reversed. Okay. So if you don't have any oxygen, so you cover your plant with the paper towel, and it's really wet, they can't breathe, they won't grow. You also need a favorable temperature. If it's too cold, nah. If it's too hot, eh. We don't use soil, so some plants will not grow without soil, and that they also depend on your plant. Most plants will germinate best around room temperature. Germination is a complex process. You get your baby, it eats the starch, and it starts to grow and to form roots, leaves, and stems. The radicle is the root where it all begins. This process usually takes 4 to 5 days, so you should see something happening today. Under cold conditions, it might take longer. So if you have nothing going on, chip your seeds, add water, see what happens. If you have something going on, take pictures of it and record the data. The direction will grow, the radicle will grow in the direction the kernel tip is pointing. Later, you have smaller roots emerge. The roots like to spread out, and we'll talk about that because the more it spreads out, the more surface area it has, the more ability it has to absorb water. This is a beautiful page, but it's mostly bonus. It's a lot of information here. I'm really concerned with the garden bean. You have a radicle, the cotyledon, your stem, hypocotyl, epicotyl. Corn, I don't really care. They're all the same thing if you look at them. Radical, hypocotyl, epicotyl, okay? So focus on the garden bean. That is your obligation. In summary, germination requires water, light, oxygen, and temperature. Look over the slides. The important slides are this slide here with seed germination, with the radical and garden bean, and going back, this page here, the germination table, pre-germination, germination, post. And then finally, I love this part, this page right here, seed stages leading to cell division, and this beautiful seed part with the parts of the seed. You're more than welcome to go back over to PowerPoint and highlight those pages. I will just print it out and highlight those 4 pages, okay? And I hope you enjoyed this conversation.